everybody. And our speaker for today is Dr. Muhammad Shaban. Dr. Shaban is a machine learning scientist at the AI for Pathology Image Analysis Lab at Brigham and Women's Hospital. This role follows his completion of over 2 years of postdoctoral research at the same lab. He earned his PhD in computer science from the University of Warwick, while focusing on medical image analysis. He has had over 8 years of research experience in computer vision, deep learning, and AI-driven medical image analysis. His primary focus is developing advanced algorithms for critical clinical applications for computational pathology. Please welcome Dr. Shaban.
The kind introduction. So I would also like to thank the organizer for giving me this opportunity to present here. So the presentations are quite diverse, like the topics are quite different. So, and my presentation is not different. It's again like it's about biology, but still in terms of like machine learning. So kind of different from the previous presentations. Okay, so the topic of my today's talk is self-phenotyping in highly multiplex imaging through machine learning. So perhaps most of the people may not know what are the highplex images or what is self-phenotyping. So in this talk, first I will give a brief background of the talk, of the topics, and then I will state the problem followed by related work, and then at the end I will present the proposed solution of the problem. So the first thing is what is self-phenotyping? So in biological term, is self-phenotyping is the process of characterization and quantification of complex molecular interaction within cell. But in simple term, or in you can say the technical term, it's just like classifying a given cell into different cell types. So the example, some self-phenotype examples are like T cells, B cells, perhaps only some of you may know these type of cells, but these type of cells are present in our body and all other animals as well. So now the next thing is why we are interested in self-phenotyping, why self-phenotyping is important. So the self-phenotyping is basically the key step to understand the tumor microenvironment. And then it also helps us in understanding the disease and drug discovery and in basic research. So now we know the importance of self-phenotyping. Now the next thing is how can we do the self-phenotyping? One of the way is using the H&E images, which are hematoxylin and eosin images. These images are gold standard for the tumor diagnosis, right? So, and they are readily available in almost every cancer hospitals. So one of the simple solution is if we want to understand, we can perhaps rely on these H&E images. So but one of the issue is these images only highlight specific type of information that we need for self-phenotyping. These images highlight the epithelium cells and immune cells, but what we really need, we really want to understand tumor microenvironment, we really need to go in depth, and we want to find where are the T cells, where are the B cells, and different other types of cells. But with these images, we cannot do that. So we need something different or something more in depth. So for this, people have developed a new technology, which is known as multiplex images. So here multiplex images just mean there are multiple layers. So we can still consider H&E images as multiplex images, or perhaps the twoplex images, where we can see the hematoxylin, which highlight the nuclear cell nucleus, and then we can consider the eosin stain, which highlight the cytoplasmic region of cell. So, but it's still like only two level of information, but we want more. So for that, there are like different imaging technologies. So one like few common technologies are like codex, maybe SciSafe and cell dive, where we can like where we can mark like up to 60 different markers. So here in this image, you can see the example, like first, like the blue and the pink image is like H&E image. And then I like overlay the like multiplex image where like the same cells are represented by different color, like the cell nucleus is represented by like blue region, and then cytoplasm is with the green, and then cell membrane with like the orange region. So similarly, like in this image, I only like showed like three, but let's say if you have like more than three, or let's say 10, 20 markers, so obviously we cannot really visualize those here. But this is how we can like see the different level of information. Let's say if we have a like CD4 marker, which basically highlight the CD4 cells. So then in that marker, we will only see the cells which are like CD4 positive. So that's the benefit of these images. Like let's say for a given tumor, if you know like different type of subcells, so then you can perhaps understand the tumor environment much better. So now the next thing is like, okay, now we have the imaging technology. Now we want to do the cell phenotyping. So how we can do like in the traditional way, the simplest option is like first they get they do the cell segmentation in these images, and then they calculate the mean value of each cell marker, and then they quantify those. Like okay, if us like for B cells, like if CD20 marker like here, like this is just an example, like if this marker is high, then the pathologist like label this thing like okay, this is B cells, and then if CD4 marker is high, then this is T cell. So this is the like usual way, like where they look at the images, they like do the cell segmentation, then they calculate the mean values, and based on those mean values, they assign labels to different cell types. But apparently like the issue is basically this is a manual step, like where we need like human input. So now the question is like, can we automate this thing, like just to like remove the issue of like subjectivity? Because when we ask this thing to do, like if a human or an expert do this thing, like there is a chance that like there will be subjectivity, and it will also be like time consuming. So one of the solution is like we can use like machine learning based method, which can like obviously get rid of the subjectivity issue and also reduce the labor intensiveness. Like with the machine learning model, we can really quickly like classify so many different cells within like maybe 1 hour, like few within like really short time compared to like a human, right? So there are like some methods, and those methods are broadly categorized in like two different broad categories of machine learning. One is like unsupervised. So in unsupervised machine learning, basically we just use the domain knowledge. Like let's say we have a marker of CD4 cells, and we know that like okay, if there are like some cells with higher intensity in this one, so we'll label them like CD4 cells. And but the issue with these models are like they are relatively less accurate, because sometimes there's like noise in those images, and then we got like lower performance. On the other side, there is another set of methods which are known as like supervised method. So in these methods, we basically give small, we can say like we give some annotated labels, and then we train a machine learning model on those like on those labels, and then we get like much better performance. But the issue with the current methods are like they are like better, but they also computationally expensive. So now what we really need, we need a method that that should be supervised so that we can get a higher performance, but it should also be like computationally less expensive so that we can quickly like do the cell phenotyping. Basically here quickly mean like we can do the cell phenotyping with less resources instead of relying on really high computational expensive like machines. So for that, we developed a lightweight cell phenotyping method where using the multiplex images, and then we evaluate the performance of that method using different evaluation metrics. And then we also compared it with different existing cell phenotyping methods. So for the development of this method, we use like 5 different data sets. So 3 data sets are coming from like in-house, like you can say like it's coming from our lab, and 2 was like the public data sets. And then the data sets are also from like 3 from like one imaging technology and 2 from the other. And also in terms of the cancer type, like out of 5 data set, 3 was coming from like one cancer type, and the other 2 was coming from 2 other cancer types. So in terms of like the data set, it's kind of diverse. Like we have different like in-house data set, external data set, and different imaging modalities and different cancer types. So this is kind of important for like to develop a robust machine learning method. So in terms of the actual data set, like in terms of the numbers, for different data set, we have like different markers. And then the reason we have different marker is basically those data set were collected part of different studies. So in each study, like they decide like how many and what type of marker they want to use. And then again, in terms of the ground truth labeling, they label like different cell types based on the study design. So that's why here you can see like the different markers have like different data sets have different number of markers and different number of annotated cell phenotypes. So to do this thing, like we do one data pre-processing step. What we did basically, we take the images and we use an existing cell segmentation method. So which basically that segmentation method like segment the cells in the image, and then we use those like that cell information for the downstream task, which is basically cell phenotyping. So here is an example of the results of the cell segmentation method. On the left, you can see a image which is kind of a duppy marker, which you can say a nuclear marker. So we can see the cell nucleus here. And then this method basically segment those regions and also like expanded a little bit to capture the whole cell. So, and then the next step, like now we have the images, and now we also have the cell segmentation information. So to simplify the problem, what we can do, like we can like list all the cells, and then for each individual marker, we can calculate the mean value. Like it's basically the same process, like how they do manually, how they do the cell phenotyping manually. So we follow the same thing. So at the end, like we have, you can say, a table where the rows represent different cells and column represent the mean value of that cell for different markers, right? We also consider like cell size as one of the features, so that like in some cases like tumor cells are usually large and the other like immune cells are usually small. So that feature may also help in better classification. So at the end, like after this pre-processing, we have like for a given data set, we have a like label data where we have one like table with K cells and N markers, and we also have for each cell we have like ground truth label. So for different data set, we have like different number of annotated cells. It again depend on the study. So for one data set, we have like over 1 million cells, but for others, we have like around 100 to 300K annotated cells. So in terms of the method, like the reason like we want to develop a really simple method so that anyone can use on their local machine without like the requirement of really high computational resources. So we develop a like simple perceptron network for cell phenotyping, which have like few fully connected layers. And then the task of the like network is to classify cell into given number of cell phenotypes in a given data set. So here is a like a visual, you can say the diagram, like the flow diagram of the method. So in the input, we have like the multiplex image. Here we call nplex because different data set have different number of input markers. So it's like we have like images, each marker represent one specific protein, and then we converted this thing during the data pre-processing. We converted this data set into a tabular format where each row is a cell, each column is a marker, and then we feed this tabular data to a multi-layer feed-forward network to classify into like C classes. And at the end, once we have the cell label, predicted cell label for each cell, we can map that label back to the images using the cell segmentation information. So, so for the experimental setup, obviously we want to like evaluate the model in a like really standardized way. So we follow a 5-fold cross validation which basically mean like we split the data into 5 different sets, and every time we use like like 4 sets for model training and the fifth one for evaluation. So, and then in terms of evaluation, we use like different evaluation metrics. Like these are like standard evaluation metrics for evaluating a classification model, which is like precision, recall, F1 score, and confusion metrics. So in terms of performance, we like our model achieve a reasonable performance. So here reasonable is basically mean where the pathologist or the end user are happy with the model performance, because we cannot really get the like the 100% performance. And one of the reason is basically the cell segmentation is not really precise. So here we achieve like F1 score, or average F1 score above like 0.8, which is kind of acceptable for the end user of the model. And if we look at the confusion metrics, we can see like the high like mapping between the ground truth labels and then the predictor classes. Like there is some misclassification between the classes, but that misclassification is generally attributed to like the biological correlation between those classes, just like CD4 and CD8. Right. So, and then there is another other way of evaluating. So basically, here you can see like two figures. On the left is the ground truth, which basically means this is the way how they evaluate the model, like when they assign the labels. So basically they again visualize this thing in this like cell expression matrix form. So where they want to like see like generally for let's say B cells, which marker is high, and then because they know like for B cells which marker should be high, so from here they can like kind of do a sanity check. So similarly, what we did, we also generated mm, this cell expression matrix for the predictor labels, and you can see usually they are like pretty much same. And one of the reason is because the overall performance is same, so that's why they are also like same. Right. So, and then as a next step, like we compare our model with the existing like supervised and unsupervised method, which are like Oster and CellCiter. Like these are just name of two different methods. So here you can see like our model outperform to the existing models. So in this setting, we basically what we did, we use these model, like we take the code of those models and we train those model on our data sets. Right. So here are some visual results. So it's like the comparison between our model, which we named as maps, and then one of the supervised mother model, which is like CellCiter. So, and then the last basically the difference where we say like we color the cells which are classified correctly by both model as gray, and then the where both models are incorrect, it's I think blue, and then the cells correctly classified by our model are like red, and then the cells correctly classified only by the other model is green. Right. So now after this, we also compare our model with one of the existing like on one of the existing public data set. And the reason we did this because we are training the existing model on our data set. So one of the obvious critique could be like, okay, maybe we are not putting enough effort to train the models, and that's why our model is performing well compared to the existing models. So what we did, we took the one of the public data set where the results are already available. So we just train our model on that and then presented the results. So as you can see, like our model still outperform that supervised model. So, and then the next thing is like, obviously we claim that like we want to develop a model which is fast and require less computation compared to the existing supervised method. So we compare the like runtime, memory usage, and then the GPU or like memory usage. And in terms of like the runtime and the GPU memory usage, our model and the unsupervised model are quite comparable. So whereas the other model, like cell citer, is like taking too much memory. So, and then the other, as a last set of evaluation, like we also evaluate like what if we don't really have like so many labels? What if we have only small percentage of labels? So then we evaluate our model using training the model only on like 5% of data, 10%, and then we gradually increase the percentage of data. So with this, we basically want to show that like model still perform reasonably well even with just 5% of the training data. So yeah, okay. So after this, we also like found some of the limitation of our models, which is basically like when we try to like we train the model and tested the model on same data set, it works well. But when we try to like evaluate the same model on some other data set, then the performance decrease. So then we also like evaluate other models and we find the similar pattern with the other models as well. So apparently it's basically the issue of generalization, and one of the reason of like this limited generalization could be these imaging technologies. Like currently these imaging technologies are not really stable compared to the other like HND images. So usually there are like so many artifacts and like the batch effect, where we call like one set of like the data generated in one setting has some different distribution, and then the data generated in another setting usually have like different distribution. So that's one of the issue where we are still working and developing like new, more generalized methods. But that's kind of update that's kind of the update on this cell phenotyping model. So at the end, I would like to like thanks my lab and my PI and also my collaborators in the Jiang lab. And I also like to thank you for attending this presentation. Thank you. Thank you, Pastor.
Learning means that like a possible solution could be like machine learning. How accurate would the machine like be and to what extent?
Oh, yeah. So in terms of accuracy, like you can look at the numbers that I presented. Is like the F1 score. It's F1 score is basically like weighted accuracy. You can say the average of precision in recall, and in this case, it's like about 80. So it in this specific case, it's like about 80, but it depends. Like if we try to evaluate this thing on some different data set, accuracy may decrease. So yeah, this is the way. But basically what we try to do here, like we try to mimic what the actual end user do. Like when they have a data set, usually they annotate the data set and then they use. So what we did, we said like, okay, you only annotate a small portion of the data set, we will train a model, and then we'll present the accuracy. Let's say in this case it's like 80. So if that thing is acceptable, then we will use that model on the remaining data set they have. So like within same lab. So in that case, it's like they can save effort, right?
That was a good presentation. Quick question. Is there any guideline available for machine learning validating software via FDA or any other agencies?
Yeah, actually, there are like even different journals have their own guidelines. Like when we publish in those journals, they usually like ask us to like provide a checklist where we want to like like highlight like the limitation of the model, and also how we actually develop the model, and like in which case this model will work and in which case this will fail. So yeah, there are like many like from different journals and even FDA as well.
Is there any biasness, like which portion of the slide you're taking picture? Is there any like?
Yeah, that's a like known issue in the machine learning. Like there is biasness at a different level. Let's say if we only train a model from one lab, then the model will be biased toward that lab. And if we try to evaluate the same model from the data of another lab, then it will perform like relatively like less, right? But it again depend on the type of data set. Like in this data set that I'm working on, it's basically a new technology, so the data sets are limited, so that's why we still have this kind of bias. But compared to this, like there is another field which is like computational pathology, where like we are training models on H&E images, and those images are available from like all across the globe. So whenever we train a model, we usually use images from like all possible public images, right? So in those models, there is no biasness at the like data center level, but there is still inherent biasness at the case patient level, like because the data, majority of the data sets are coming from the developing countries, right? So the model tend to perform well on like kind of white population, because the samples are coming from the white population compared to the other, or like model tend to perform better on like a developed patient from the developed countries compared to others. So there are like different level of biasness, so it depends like what type of data we have.
Thank you so much. I have a question. Is this kind of like DNA sequencing?
Like this model.
Yeah, the data sets and stuff.
Yeah, this is a, like a proteomic data set. So basically what the difference is, like in DNA, we have like we don't really have spatial information. So in this case, let's say if we have a tissue sample, we have like different cells. So we have like kind of cell level information, like whether this cell is positive for a specific marker or not, right? So that's the plus point of this data set. It basically introduce the spatial information. Although in like in this work, we haven't really used that spatial information, because we just like convert this data set at a cell level. But in another work, we are working on that. Right.
Thank you for the information. I have actually two questions. One is related to, already you mentioned regarding the passing. So, what is the balancing regarding the classification? So, how to have actually address the balancing of different classifications?
Oh, okay. So you are talking about the class imbalance, right? Yeah. So yeah, like there are different techniques to address this issue. One is like oversampling, undersampling. So in this case, basically we rely on oversampling. Basically, while model training, we feed the samples from the like with less samples, we feed more instances of the sample while model training, right? So let's say if a class have like 1,000 a sample, another one has 100. So while each model iteration, we sample more from the class with 100 samples. So it's kind of at the end, it's like a model see a balanced number of sample from each class. But this is like a known issue, and it depends. Like let's say if there is a like huge class imbalance, like one class have like let's say 10 or 20 cell, another one have like 1,000 or 2,000 cell, then it will be difficult to address. Then the best solution is to like get more data. But in this specific case, we do have like class imbalance, but we handle this thing with oversampling.
Okay. And second question is actually like when you explain the scores, did you apply any kind of um, explainability or any interpretability of the machine?
Right. So yeah, there is a way in this in these models, like we can do the interpretability analysis. So we did one like one type of analysis. So that was like I can show you. Yeah. So like those results are here, but what we did basically, we for each like cell type, we figure out that like how much like attention, or you can say the how, model focused how much focus model gave to each individual marker. So let's say if here in this case, if we see that like if the cells are B cells, So model should give more weightage or more focus to the marker which are related to the B cells. So we did this analysis. So it was not really perfect because that interpretability is basically it's not like well known, like well interpretable. One of the reason is like these models are kind of black box. So this is just like one way to kind of explain it, but we can't really 100% guarantee that like, okay, this is the way model is actually predicting. It's just like one way. So we did that, but it was like not really perfect. But
Yeah, there was a like deep shadow.
yeah, that's basically what I use. Uh, I was not expecting that many questions.
So I was wondering, are there ways to improve the accuracy of the model?
Yeah, obviously there are like pretty straightforward ways to improve the performance. One of like in this specific case, like basically we need to like figure out like how can we improve. One obvious way is like let's say if we give more data, right? So then there is like chances that like we will get a better accuracy. Like in these days, perhaps you have heard about like different foundation models, these llama or like language models. Those are all on trained on like millions of like images or patches, right? So one obvious way is like adding more samples, and then the other thing is like in this specific case is basically we are relying on automated cell segmentation, right? Which basically mean like if algorithm has segmented a cell incorrectly, then we cannot really like this model cannot really do anything, right? Because the if the cell segmentation is not right, which basically mean the mean cell expression is not right, and which basically mean like algorithm will just use the mean value, which is not right. So it will predict incorrectly, right? So, but these are the few things that we can like focus on and increase the model performance.
I really liked your presentation. So my question is, this year, like there was a Nobel Peace Prize for the studies of machine learning and predicting the protein structures. Are you familiar with this? And is your algorithm similar to this one?
Oh, okay. Yeah, I'm familiar with that. And it's kind of similar to that only in term of like, because those models are machine learning based or AI based, and my model is also machine learning and AI based, but obviously it's not directly related to that. Those are about like protein, like structure prediction. This one is a different kind of imaging technology and different way to analyze or like understand the tumor microenvironment. Like the data set is different. So yeah.
Thank you.
Okay.
Thank you. This is really a top, very promising technique, and I think I was really familiar with this technique. Now that we know the value of RNA sequencing, single cell RNA sequencing, it's really published everywhere. We're in that top channels. Eventually, I can probably foresee your technique will reach that level of doing multiplex of different markers, and not just one single marker, but probably combination of the extent, the quantification reach. So probably more finely differentiate some of the cells, even within the same operation. And
Right.
if we could see that kind of big picture, how far away are we from reaching that ultimate goal of using already fixed and corrected samples? What do we have to do in order to be achieved?
Okay, so if I'm understand your question correctly, you are like saying that like when we will reach like in this multiplex imaging, when this multiplex imaging domain will like reach the level that the
Right
RNA
now, I
sequence.
can see if you specifically discuss one marker, another marker separately, the score really seems to be very promising. And eventually, probably you'll be wanting to combine them and doing a layer quantification to more finely tuning the differentiation. So even within the macrophage population, even within that T cell population, because there is a gradient spectrum of expression.
Right. So yeah, like currently we are developing like the model where we consider like multiple markers simultaneously and like that's even in this case like we consider multiple markers for cell phenotyping. Although we can perhaps train model individually for each individual cell, we can only rely on like the specific markers. But currently we are working on a model where we basically pull the data set from like different labs and different publicly available multiplex data sets, and we are trying to build a one kind of foundational model which will be used for like the downstream task. Let's say if we have enough like patient samples, let's say 1,000 samples with this type of data sets, right? So then it will be like instead of like manually diagnosing or like manually finding a different patterns, we can use like those foundational model features and the survival data, and we can automatically find those like why this patient have a poor survival or a better survival. So we are going toward that direction. But one of the like there are like limitations. One is like this technology is quite expensive, right? So usually the number of samples are limited per like study. But overall, it's like there is a like growing interest in this field. And even like this year like the nature method basically consider this technology as the method of the year, right? So which basically mean like there is a good future for this domain.
Have somebody, for example, let's put it a discussion with macrophages, like firstly, you have N1 macrophage, CD80, CD206, and so on. Even if you look at the CD86 first operation, there seems to be really low operation.
Oh, yeah.
Have you detected such things using this data already, or somebody else?
So yeah, so actually I'm working on kind of the machine learning part where I basically my focus is to just like maybe classify or present like this data set in a way that's like easy to use for the actual biologist or actual like basic life science person, because they do these kind of analysis. Like they do like, okay, like if let's say there are like 10 macrophages, or what type of macrophages may have like good impact or bad impact or patient prognosis, right? So that's basically like each individual question is like one research direction, right? But my focus is basically developing on AI models that can be used by all those, like, can be used for all those questions, right? So the idea is like to develop some models that people can then use for each and to address each individual question, right? Yeah.
Well, thank you. It's really interesting.
Thank you so much. You have this.
Great presentation, thank you. We did ask what kind of library did you use for this kind of model? And also, I was wondering, like, the data set that you're using to train the model are based, I guess, on segmentation and validation performed by humans. And do
Yeah.
you think that the model accuracy would be comparable to the, for example, the bias between annotation, which seems that would be allocated the same data sets?
Right.
Maybe the error, like, range would be similar to the accuracy of the model.
Right. So for the first question, like we use like PyTorch based libraries, like these are deep learning libraries to train these models. And then the second question is about like if we do the prediction on a new data set and we ask a like two different pathologists to do the annotation, and then we compare the results, like the agreement between those two pathologists and then our model. We haven't really did this, but this is a like known thing, and there will be like high variation between two pathologists. And why I'm saying that, like in this case, if you look at the numbers, like for one of the data set, we have like 1.5 million cells, right? Which basically mean those cells are not annotated manually, right? That's kind of not possible, right? So what they basically did, they only annotated some. So basically they have like these markers. So they kind of adjust the threshold for each individual marker for a given image, let's say if it's a CD4 marker, they adjust the threshold, and then all those cells above that threshold, they will say that like, okay, these cells are like CD4 positive, right? So obviously that thresholding is not ideal, right? There will be like, maybe there are some cells which are high just because of some imaging artifact, right? So there are so many things, but this is how they usually annotate, and that's one of the reason like I have observed, like I can only get the accuracy at a certain level, right? Because then after that, it's not possible. Let's say maybe my algorithm is correcting a cell correctly, but just because I have a like incorrect label, I will consider that thing a misprediction, right? So that's kind of one of the issue that I have faced in this project. Okay, thank you. Thank you, everyone.